Hi everyone, my name is Kaori Yamada. I am an assistant professor in the Department of Pharmacology and Ophthalmology. We are studying vascular function in cancer metastasis and eye diseases. The potential projects in our laboratory are the mechanisms of how uveal melanoma escapes from the eyes to metastasize to the liver, the mechanisms of how breast cancer invades the brain through the tightest barrier of the blood-brain barrier, and the mechanisms of subretinal fibrosis development in neovascular age-related macular degeneration. Endothelial cells form a barrier to protect the tissue. The endothelial barrier is formed by adherence junction and tight junction between endothelial cells and endothelial cells. Pericytes protect endothelial barrier by adhering to endothelial layer. In the case of blood-brain barrier and blood-retinal barrier, astrocytes further protect the barrier integrity. Cancer cells secrete factors to induce vascular leakage so that cancer cells can transmigrate through the endothelial barrier. Two of our projects are to identify factors from cancer cells and the mechanisms how endothelial cells respond and react to the factors in cancer cell transmigration. One of such factors, and the most famous factor, is VEGF, vascular endothelial growth factor. VEGF induces pericyte detachment and barrier relaxation that increases vascular leakage. It also induces angiogenesis, the formation of new blood vessels from the existing vasculature. In tumors, angiogenesis supplies nutrients and oxygen, supporting tumor growth. In patients with ocular melanoma, higher expression of VEGF-A is associated with shorter progression-free interval. Ocular melanoma is also called as uveal melanoma, developing at the uvea in the eyes. Uveal melanoma is highly metastatic and spreads through blood vessels because there are no lymphatic vessels inside the eyes. We previously showed that uveal melanoma cells secrete VEGF to induce endothelial permeability. Human retinal endothelial cells are seated on the top of biotinylated gelatin, and cells are stimulated with either VEGF or conditioned media from uveal melanoma monoculture or uveal melanoma and endothelial co-culture, with or without aflibercept, that is anti-VEGF treatment. If there is leakage, biotinylated gelatin is exposed and streptavidin can bind to it, so that it is shown in red in the figure. As a positive control, VEGF induced endothelial permeability. Similarly, conditioned media from uveal melanoma and endothelial co-culture induced endothelial permeability. Such permeability was inhibited by aflibercept, anti-VEGF treatment. This data shows that uveal melanoma secrete VEGF to induce endothelial permeability. We tested 3 uveal melanoma cell lines. All of them induced permeability by secreting VEGF. Then we tested transendothelial migration of the cancer cells using organ on a chip. This mimics cancer cell intravasation from the tissue to the circulation. We put M-cherry expressing cancer cells and collagen gel in the middle lane. Then we put blue-stained endothelial cells in the next lane, so that endothelial cells form a barrier on the interface of the collagen gel. Then we monitored cancer cells transmigration through the endothelial barrier. In control, cancer cells transmigrated through the endothelial barrier to the next lane. Chi is a peptide that we developed to inhibit VEGFR2 trafficking to the cell surface, thereby inhibiting VEGF-induced signaling in endothelial cells, such as angiogenesis and vascular leakage. Chi prevented the transmigration of the cancer cells. Similarly, anti-VEGF also prevented cancer cell transmigration in 2 out of 3 uveal melanoma cells. However, one very metastatic uveal melanoma cell lines seems to have VEGF independent mechanisms to metastasize. To examine how uveal melanoma affect gene expression profiles in endothelial cells, we did co-culture of human retina endothelial cells with uveal melanoma cell line. Isolated endothelial cells using CD31Bs and performed RNA sequencing analysis. Gene ontology analysis of the differentially expressed genes in endothelial cells with co-culture with uveal melanoma showed several pathways, including cell-cell adhesion via plasma membrane adhesion molecules. We focused on this pathway as it has a high gene ratio, a low adjusted p-value, and a reliable gene count. The focus is on which adhesion molecules are upregulated in cancer cells and endothelial cells to contribute to cancer cell transmigration and metastasis. Using publicly available RNA-seq data of primary uveal melanoma cells and metastasized uveal melanoma cells, and our RNA-seq data of endothelial cells, we did ligand receptor analysis. These are the ligands upregulated in metastatic uveal melanoma, and these are the genes affected in endothelial cells by uveal melanoma co-culture, either upregulated or downregulated. Based on these candidate genes, this project focuses on investigating the mechanisms of how uveal melanoma escapes from the eyes to metastasize to the liver, which adhesion molecules are highly expressed in metastatic uveal melanoma, which molecules on endothelial cells are the binding target of the adhesion molecules on uveal melanoma. How do they affect vascular permeability and transendothelial migration? The potential rotating students will use cell culture, quantitative real-time PCR, siRNA knockdown, adhesion assay, and transmigration assays in vitro. The next project is breast cancer to brain metastasis. To enter the brain, circulating cancer cells need to pass through the blood-brain barrier, which is the tightest barrier in the body. We isolated metastasized breast cancer cells from the brain after intracardiac injection. We repeated this cycle again to select brain metastasized cancer cells. Then we analyzed secreted proteins from parental breast cancer cells and brain metastasized breast cancer cells. We identified the molecule that is highly expressed in brain metastasized breast cancer cells and confirmed that by Western blotting. These are unpublished preliminary data, so let me put a black box here. This molecule is known as a serum protein secreted from liver in healthy people. The molecule from human serum did not increase endothelial permeability. However, a cancer-derived molecule significantly increased endothelial permeability. We further found that the cancer-derived molecule has a different glycosylation pattern from the serum protein. In this project, studying the mechanisms of how breast cancer invades the brain through the tightest barrier of the BBB, we will identify how the expression of this molecule is regulated in metastatic breast cancer cells, how its glycosylation is regulated in metastatic breast cancer cells. The potential rotating student will use in vitro cell culture, siRNA knockdown, qPCR, and Western blotting. Another project is subretinal fibrosis development in neovascular age-related macular degeneration. In neovascular AMD, upregulated VEGF induces neovascularization of choroidal blood vessels, which are leaky and cause inflammation and damage to the retina. Anti-VEGF is effective in inhibiting vascular leakage and angiogenesis, but the patients still lose vision due to the subretinal fibrosis development. Subretinal fibrosis is a deposit of extracellular matrix that is secreted by myofibroblast. The origin of myofibroblast can be retina-pigmented epithelium, endothelial cells, pericytes, and macrophages, through their mesenchymal transition. As a neovascular AMD model, we use the laser-induced colloidal neovascularization model. After laser burn on Bruch's membrane, vascular leakage is observed on day 3, and colloidal neovascularization is observed on day 7 to 14, which mimics the disease progression of neovascular AMD. We monitor mice with optical coherence tomography and fluorescence angiography. We can also monitor subretinal fibrosis development by inducing laser burns twice at 7 days interval and monitor mice longer, up to 40 days. We did RNA-seq analysis of mouse eyes from healthy control and mice with subretinal fibrosis without any treatment, or with anti-VEGF or Chi treatment. We identified several differentially expressed genes in subretinal fibrosis. Among them, endothelin 2 was further upregulated after anti-VEGF treatment. In this project, studying the mechanisms of subretinal fibrosis development in neovascular AMD, we will examine whether endothelin 2 induces epithelium mesenchymal transition and endothelial mesenchymal transition, and whether endothelin 2 affects endothelial permeability and leukocyte transmigration by using an in vitro system. Thank you for listening. If you are interested, please feel free to contact me. Thank you.